Okay, so again, Assalamu alaikum and good afternoon to everyone to yet another webinar from Omics Nexus. So today we will be delving into the Bioinformatics Starter Kit. So it will be your first step with Omics Nexus into the world of bioinformatics. It is for the whole beginners who are just starting or transitioning into their careers to bioinformatics. So before moving on, let me just give you guys a little introduction of myself. So I'm currently, and my name is Taimur Sikandar, and I'm currently an ambassador of Omics Nexus, and I'm pursuing biotechnology. And in my last year, uh, in my undergraduate studies from University of Veterinary and Animal Sciences. So moving on, let me introduce you guys to our organization. So what is basically Omics Nexus? So we are basically making bioinformatics accessible to everyone in the field. And we are redefining what it actually means to be a scientific solution partner. We go beyond the typical service model, and in comparison, we have deep expertise in expert bioanalysis, bioinformatics, NGS data analysis, pipeline development, biomarker discovery, and on top of that, we do deal with the systematic reviews, analysis, and scientific writings as well, and in accordance with the healthcare business development to deliver data-driven impacts. Okay. So what we actually offer in this organization. So we are delivering advanced bioinformatics and AI solutions that drive faster, smarter, and healthcare decisions. So we are providing, as I have already discussed with you guys, that we are providing bioinformatics analysis on various genomic data. So we are providing AI and machine learning services to build basically AI and ML models to train precision data and to build your genomic models. And we are also doing the automations and pipeline developments from the start, from data collection to the interpretation and visualization, along with the statistics and data analysis of the data. And to make scientific communications even more reliable, we are also doing content drafting and illustrations as well. And will also be helping you in the research project developments. And the one thing that actually sets us apart from all of this is that we are providing you guys consultation services and listening to your goals before we actually move on to the actual projects. So, and to cope up with that, we are also providing trainings and workshops for the whole beginners to get you guys equipped with the basic knowledge of bioinformatics to went from basic to the advanced level through our training and workshop programs. Sorry. Okay. So what actually sets us apart from the other organizations? You might be wondering what sets us apart. So first is our multidisciplinary expertise. We bring together bioinformaticians, AI specialists, and statisticians under one roof. So we have like experts in every regard when it comes to bioinformatics. And we have built tailored end-to-end solutions customized for your specific data sets and objectives. And as I have already discussed, we are providing free consultations always. So please visit our website and sign up for your demo consultation so you can like avail our services. And other than that, we have a 99% client satisfaction rate. We have proven results and client trust. And finally, we provide always 24/7 support to ensure your deadlines are always met. Moving on. So let me introduce you guys to our team, which is our core team that have established this organization, which is called Omics Nexus. So first off, we have our Chief Executive Officer, Sir Nadeem Khan, who is also a bioinformatician. He has done his master's from National University of Science and Technology, Islamabad, and he is specialist in NGS data analysis, multi-omics, bioinformatics project development. Moving on, we have Sir Hazrat Moaz. He is Chief Operational Officer and is also a bioinformatician. He's currently pursuing his master's in bioinformatics from us as well. And he's specialist in applied AI and ML models, bioinformatics data analysis, web development, and IT. And then we have Munib Nasi. He's Chief Technical Officer and is also have pursued his master's from NUS as well. He's specialist in bioinformatics pipeline development, structural bioinformatics, and of course, consultancy as well. Lastly, we have Sir Haseem Mansoor, who is Chief Scientific Officer in the Omics Nexus. He has done his master's from NUS as well and is currently doing his PhD from Italy. And he's specialist in metagenomics, ML-based biomarkers discovery, and scientific communication as well. And let me introduce you guys to our free webinar series that we have conducted since we have started this organization. So first off, we also conducted the Bioinformatics Starter Kit to familiarize the audience with the basic concepts of bioinformatics, and we went to the AI and bioinformatics in modern healthcare because, as we know, AI, artificial intelligence, and data science holds top importance in the field of bioinformatics. And then we had our webinar in the previous month on the Git and GitHub, where Sandeep Nasir specifically introduced us what is the difference between Git and GitHub, and introduced us to the commands, basic Git commands to use on Linux. And just recently, we conducted a comprehensive webinar on the RNA-seq data analysis, in which our own Sanadeem Khan told us that every and each step which goes into the pipeline development from the data to discovery into the bioinformatics, into the RNA, using the RNA-seq data. Okay. And apart from these webinar series for the thorough and detailed training of bioinformatics, we have also conducted the workshop series as well. So first off, we conducted 2-week crash course led by our son Nadeem and Sir Haseeb as well, in which we covered from introduction to metagenomics and Linux to the data visualization in R. And next, just recently in this past month, in January, we conducted a workshop series on the transcriptomics-driven data, which was part of 3 workshops combined. And we used microarray data along with RNA-seq data to actually deal with the lung cancer and the brain tumor classifications as well. So I actually took part in this workshop as well. So I can provide a better review. Okay. And after this, what are the milestones that we have so far achieved? We started our ambassadors program in the previous year, like in October, yeah. And we expanded our outreach program to many institutes across the nation. And now we have ambassadors in different institutes delivering our outreach program. So it is one of our big fit. And I also applied in this, so I'm the ambassador right now as well. And after that, we upgraded our system to the high performing computing power and to actually deal with the big data analysis so that we can provide actually good services to the clients using their big data so that we can work efficiently. So you guys can trust on us. And other than that, now our website is live. So go sign, it's omicsnexus.org. So go sign up to our newsletter for the updates and all, and what our upcoming events will be so that you can stay tuned with us. So let me just introduce you guys what we have upcoming plans in our future in Omics Nexus. So first, we are planning to have a single cell RNA-seq workshop in which we will be providing you guys with the knowledge of single cell data generation and preprocessing and end-to-end analysis with SCAMP and cell type annotation and differential expression as well. And after that, we're planning to have a bioinformatics summer school, which will be totally from beginner level to the advanced level to familiarize yourselves and to the students who are actually from biology, biotechnology, or microbiology background who don't have much knowledge of coding and programming to be familiarized with the Linux, Python, and R programming to the efficient level. And we will be dealing with human genomics, structural bioinformatics, omics foundations, and microbial genomics as well. Okay, so let us start our session by welcoming our guest speaker for today is Professor Dr. Ibrar Khan. He's an associate professor and director of Center of Biotechnology and Microbiology from University of Peshawar. He has successfully supervised numerous PhD and MPhil scholars and led multiple funded research projects as principal investigator and co-principal investigator, reflecting his significant contributions to research and higher education. And we are truly honored to have him in our session today. So over to you, sir. And meet our speaker for today. And again, I am introducing my mentor as well since I joined the Omics Nexus, Nadeem Khan, who is an expert bioinformatician with an MS in bioinformatics from NURSE specializing in NGS data analysis using Bash, Python, and R programming. And he has a strong focus on multi-omics, data integration, and contributed to core tool development and applied research projects. And he has an expert level skill on the R programming and Python, and I'm telling you guys with experience. So yeah, we are truly honored to have you with us, sir. So over to you.
Perfect. Okay, Bismillahirrahmanirrahim. Assalamu alaikum. Hope you all are fine. And thank you very much for joining this webinar of Bioinformatics Starter Kit. As Temur have mentioned, I'm Nadeem Khan, the founder of Omics Nexus. And Omics Nexus have a single and a unique motto of making bioinformatics accessible to everyone. So this webinar is related to the beginners or who wants to pursue their career in bioinformatics, or they want to integrate their research, the bioinformatics into their research. So we'll be dealing with it. Okay, so the objectives, the contents of today's webinar is the introduction, what is bioinformatics, uh, explaining bioinformatics through practical examples, careers and trends in bioinformatics, and how biosciences students can integrate bioinformatics into their research. I'll be explaining it that if a person is from microbiology or biotechnology or biochemistry, so how they can start their career and how they can start their research in bioinformatics. Then we're having a starting with bioinformatics roadmap, that how you will start from the scratch. And lastly, the most important thing is the question answer session. All are requested to post their queries in the WhatsApp group. In the last of the session, I'll be answering each and every query, inshallah. Okay. Okay, so first of all, the main objectives of this webinar is that to give clarity and confidence while choosing the right career journey. And we do have some intermediate students, so it's also a type of career counseling session also, that how we can start with the bioinformatics. And the other most important objective is that how bioinformatics is reshaping research and how the biosciences students can start their career in bioinformatics. So these are the main two objectives. So we have two scenarios here. Current scenario, some of you are looking ahead wondering which career to choose, the intermediate students, and the majority of the students are already in biosciences wondering how bioinformatics can fit in. So we are having two scenarios and we'll be addressing these two scenarios parallelly. Okay, so this session connects both the journey. So let's start. Most important thing and the very basic thing is that what is bioinformatics? I think most of the students, the participants should know that what is bioinformatics. But for me, I define bioinformatics in very simple terms. It's an interdisciplinary field. Uh, Three fields combined to make this specific field. Like the first field is biology, the second is statistics, and the third is computer science. If you have knowledge of these three fields, you can be a better and you can say a better bioinformatician. So you should have these three knowledge, these three domains in mind: biology, statistics, and computer science. Now, most of the stuffs can be easily understandable by giving example that how biology, statistics, and computer science are combined together to form a bioinformatics. So I'll be explaining it to an example. Let's start with the biology. In biology, if we see that we all know that not all genes are active at the same time. For example, the insulin gene is active in pancreas cell, but not in brain cell. We know that there is a specific type of gene regulation. Okay, we have 200 types of cells inside our body, and yes, we do have 46 chromosomes inside it. But not all chromosomes are expressed. Not all genes are expressed in that same cell. Okay, each cell have their own expression system. So in biology, we have the expression of different genes. So this is basically known as this is called as gene expression data. Now, if we want to look into that, how if we are having 100 individuals, okay, and we can say that some individuals are healthy individuals and some are, we can say, diseased individuals, and they have different type of gene expression data. So if we want to compare thousands of genes across hundreds of patients, it's impossible to check it manually. Okay, so what computer science do is that it provides us a platform to analyze our data. It provides different softwares, different tools to process that huge amount of data, because computer help us read and store these massive tables of gene activity like Excel, but as much bigger, as faster in scale. Okay, so the data comes from the biology part. Computer science provide tools and softwares to analyze the data. Okay, now here comes the statistics. Statistics help us find which genes are more active in the disease patient and in the healthy individuals. Okay, so here basically, if we give an example that, for example, we have a cancer gene is always switched on in patient but not in healthy people. Statistics confirm that this difference is real and significant. Let me explain it in a very easy way. For example, we are having plenty of genes. For example, we are having two individuals, and inside the two individual we have 20,000 genes expressions. Okay, now we want to find out that which genes are significantly upregulated or downregulated. So dealing with those all genes do require some softwares and tools and some, we can say, automated pipelines to analyze the data, because it will not be manually analyzed. So computer science provide the tools and softwares. Now we are generating a hypothesis that, for example, which genes are upregulated, which genes are downregulated. Here comes the statistics, the mathematics, that uh, the genes that are significantly dysregulated, it do follow some statistical rules and formulas. So here the statistical helps us out. So bioinformatics is helping us in understanding the gene regulation, dealing with the massive data, plus also it helps us in generating statistically significant hypotheses. So this is an example of bioinformatics. Let me give you another example of that. What is basically bioinformatics? Now, we all know that mutations are small changes in DNA that might change how a protein works. We all know that what is mutation, and sometimes it leads to a disease. Okay. Now, how computer science fits in? From the biology, we get a lot of genomic data, a lot of DNA type of data, the nucleotide sequences. We get it from the biology. Okay. Now, what does the computer science do? We all know that a single human has over 3 billion letters of DNA. And to find one mutation, we need computers. Okay, we need a lot of high performance computers and tools and softwares to check that where is the mutation inside these 3 billion DNA letters. Okay, so computer science help us in providing the tools and the software. Now here come again the statistics that, for example, now imagine we found that same mutation is in 20 out of 100 patients, and that mutation is also present in 1 out of 100 healthy people. Now same mutation but have different proportion. Now we have to generate a hypothesis that either this mutation is significant or not, or either it is a random mutation, or either it is causing a disease. So here comes the statistics. There are different formulas, different other type of mathematical models that help us to generate a hypothesis that yes, this mutation is significant and to be analyzed further, or not, this is just a random mutation. So here the data come from the genomic data, come from the biology. Computer science help us to analyze that huge amount of data, and statistics provide us different formulas, different thresholds, and different statistically significant, we can say, analysis that to tell us that yes, this mutation is significant or not. So this is basically known as bioinformatics. There are a lot of different examples that can easily explain that what is bioinformatics, what bioinformatics is, but we can say that knowing these three domains can help us to become a better bioinformatician. Okay, so you can now compare yourself that you know what is biology better enough. Then you can concentrate on the computer science. That you should work on different tools and software. Then I'll be explaining it. And statistics, we are dealing with the statistics from the day first when we were in our we start our education. So basically we should also know that how statistics deal and what does statistics mean. So these are three domains. Okay, so at least you should have commands over these three domains. Now, why bioinformatics matters today? If we can say that why this webinar is very much important for you people, and how bioinformatics is important in modern research and nowadays. So you know the COVID-19 story, okay? Before bioinformatics, I told, if I mentioned that there were a lot of diseases, a lot of different infections that took a lot of years to be tackled and to be to search that what was the causative agent. But how bioinformatics helps in tackling the COVID-19 in a very limited time. How the bioinformatics came handy within weeks. Scientists sequence SARS-CoV-2 using bioinformatics. Okay, so the genome of the COVID-19 virus was sequenced, and after that it was analyzed. Okay, it was analyzed using bioinformatics. Genomic surveillance revealed mutations and variants in real time. Okay, so we were hearing as that a new variant arrives and a new variant arrives. This was all because of the bioinformatics that they were basically dealing with that specific data. When it was generated by the patient, it was analyzed and we could find out the variations that yes, COVID-19 have changed. There are mutations in it. And what happens that when after getting the real positive agent, we start manufacturing and designing vaccines in the form of multi-epitope vaccine, designing the messenger RNA vaccine. So basically, there was a wet lab part, but that wet lab part was being paced up by the bioinformatics. Okay, there were no just like that we could say that just we were generating assays and we were looking at what was the causative agent. Bioinformatics was helping it that to pace up that whole process. It was giving predictions. It was giving important data that yes, this may be the cause of this disease, and just because of this bioinformatics, things get paced up, and we could say that yes, COVID-19 issue was overcome in a very limited time. Okay, so here this is why bioinformatics matters today. Now, most importantly, bioinformatics turn data into hope and hope into life-saving action. We all know that what happens in the COVID-19. Okay. Okay. So why bioinformatics matters today again? If we could say that why we are dealing with bioinformatics, so there is a huge amount of data being generated. Okay. We can say that in 2020, just look here to the amount of data that has been generated. And we all know that bioinformatics deal with biological data. So the larger the biological data, the more we need the bioinformatics analysis. Okay, so due to the increase of genomic and the biological data, we need bioinformaticians. Look here, a single patient produces 80 plus megabytes of medical data every year. Okay, it should be analyzed, and who will be analyzing this data? Bioinformaticians will be analyzing the data. If we look here to the medical images, now we are working over medical images. You can say that you go to the hospital and you perform an MRI or CT scan. That data matters for us. We look to that data in a very greater way, in a very different way. That we find out the variations and we find out the patterns of that the medical images, that what is happening inside the body. Okay, so that data is very much important for us. Bioinformatician deal with it. Okay, so the larger the data produces, the more we need the bioinformatics the analysis and the experts. Okay, if we look to the genome registries, the genomic data, so sequencing data is being generated in a very huge pace. Okay, and that sequencing data should be analyzed. So bioinformatician will be analyzing the data. A lot of other data is being generated. In simple sense, if I can say that biological data matters a lot. So if you have biological data, you have important we can say knowledge of computer science and statistics, you can better analyze the data and generate hypotheses. Now moving towards the market growth, so we can say that from 2018 to 2025, just look here to the data being generated in the healthcare. Okay, it's more than all the other data. Bioinformatics market is set to reach is, just look here to the huge amount of the billions that how it is helping out. Okay, now International Journal of Research. This is a report that 35,000 published works in bioinformatics. Okay, a lot of research papers are being published in bioinformatics because this field is new. It has no saturation, and we have a lot of data. So the more the data, if we have more data, we have a better bioinformatician. So yes, he can generate essential hypothesis. And yes, if he generate essential hypothesis, so it should be published. And look here to the publication, the number of publications, it's huge. Okay, and bioinformatics programs it have increased to 31 folds. Okay, so this explains that why bioinformatics matters today. Now, there's also some different stuff that can explain that what why bioinformatics is important. If we are dealing with the pharma industry, so drug discovery is very much important field of bioinformatics. Academia, you all are from the academia, I think so. So at least half of the participant, uh, you join this webinar just because you know that bioinformatics is important in academia also. Also, we can deal with the land and animal conservation, public sector, biotech industry. These all have different positions of bioinformaticians. Okay, and look here to the average salaries of bioinformatics jobs. So it's quite impressive. These are the salaries. You can check it out. Okay, these are taken from published sources and reliable sources. Now, till now, where do we stand now is that some are choosing their career and the others are, we can say that they are confused. Students are confused when they perform their intermediate. So they are thinking that where we can go and what type of stuff we can acquire. So I can say that bioinformatics is a better choice for them if they want to pursue their career other than the medical field or the engineering field. Bioinformatics is also a medical field, plus it's also an engineering field. I'm telling you, okay, and it have a lot of scope. The most important participant list is that others are already in the biosciences, wondering how bioinformatics fits in. So let me explain that if you are from microbiology or biotechnology or biochemistry, how bioinformatics is going to help you in your research. So let's start with the microbiology. I am also a microbiologist. I have done my bachelor's in microbiology, so I can, and then I have shifted towards the bioinformatics, so I can explain it well that how microbiology fits in. And I think so that we have some students of microbiology also. So no need of worry, you can easily get into bioinformatics, as I have also get into bioinformatics. Look here to the metagenomics. It's a research type explanation. Metagenomics is basically when we start dealing with the microbial content of the living inside the living organism or the environment. We all know that we do have a lot of microorganisms, microbiota inside our body, and it is being proved that they have a very significant role inside our body. Okay, so let's suppose if we start analyzing the data of our microbial content of our microbiota. For example, a patient or we can say that a colorectal patient. Uh, uh, we can say that if we start analyzing the microbial data of a colorectal patient, so what will happen is basically that we will start analyzing that the gut microbiome of it. Okay, so we can check out that how the bacteria inside the colorectal cancer patient is behaving, and we can just generate hypothesis that either this microbiota is contributing in the onset of colorectal cancer or not. different hypotheses being generated. I have done a lot of research on metagenomics, okay, in my master's level. So we do have a lot of hypotheses, and we have generated a lot of hypotheses related to research type of things, okay. So metagenomics is very much important. If a person is in microbiology field, he can just start dealing with the metagenomics, because I can say that metagenomics is just like computational microbiology, okay. You are not dealing the bacteria inside the lab, you are dealing the genome of the bacteria on the computer screen, okay. This is known as metagenomics, and how microbiology students can start their career in in bioinformatics by choosing the field of metagenomics, okay, and by starting their research in metagenomics. The other thing is the vaccine designing. This is most important, and I think so that most of the people should know about it. That what is vaccine designing? That, for example, if we know the virulence factors of the bacteria that causes the disease, so we can design multi-epitope vaccines. Okay, there are a lot of databases, a lot of AI models that can predict that yes, inside this virulence factor, this protein, there are certain portions that can be epitopes that can be recognized by the B cells and the T cells and the immune cells. They can choose those epitopes, they can design a vaccine, and they can say this, yes, that yes, this vaccine has the potential to combat or to evoke the immune system of a living organism before the bacteria or the virus attacks. Okay, so you are dealing with a microorganism in the metagenomics plus in the vaccine designing, but on the computer screen, not in the lab. So you can shift your research directions into bioinformatics. I'm not telling you that you are leaving your microbiology. You are just doing some modern research. I am not telling you that wet lab is not important. It is important, but here in Pakistan, I'm telling you that we do not have better or more resources to deal with the high-end wet lab experiments. So yes, having the knowledge of bioinformatics, you can start your advanced research in microbiology, and you can publish papers even in your master's level and in your PhD level and even in your bachelor's level also. I have done it. Okay, so I'm telling you that if you have the knowledge, you have the data, you have a better supervisor, you can start your career in microbiology, shifting to bioinformatics. Okay. Now we do have some biotechnology students also. So how biotechnology fits in? There are a lot of different other examples also in microbiology, but just for the time constraints, that is why I just mentioned two examples. Now biotechnology. I have also mentioned some gene expression in the beginning. Let me tell you that, for example, we are having normal tissue and cancer tissue, okay? And we do have gene expressions. We have expressions of all the genes present in the normal tissue plus in the cancer tissue. Now we want to find out that what are the genes that are upregulated and downregulated, and what are the genes that are very much upregulated in the cancer tissues with reference to the normal samples, or they are downregulated. And so we can generate some hypotheses, and we can just find out some biomarkers that are related to, or that are used, or we can say that they are causing the disease, the cancer. Okay, so such type of research can be done in the biotechnology. If you have the genomic data and you have the skill to analyze it, start your research in biotechnology. Do not wait for the wet lab part. Okay, just get the data. We do have a lot of data in the databases in the NCBI, ArrayExpress, or we can say European Nucleotide Archive. A lot of genomic data is present. The expression profiles are present of very advanced diseases. You can retrieve it. You can start analyzing it. Okay, and it is publishable. Using secondary data can be publishable because you are dealing it with in a different angle. Okay, so biomarker discovery using gene expression profiling is an important part of biotechnology research. Now we do have a lot of other research options also. Whole genome analysis. If we want to check the entire genetic content, the DNA of the or the exomes of the patients, the cancer patients. Okay, so we can find out a lot of things like variant analysis. Okay, by finding out mutation. That, for example, if we have a gastric cancer patient and we have the data of that gastric cancer patient, the genomic data, so we can just think about it that what are the mutations that are present in this data, and by finding out that mutation, performing some statistical analysis, we can generate hypothesis that this mutation was the main cause of this, or the potential cause of this disease. Okay, so you can deal by being a biotechnology student. You can start career in bioinformatics. By analyzing the data, epigenomics is also very much important. The gene regulation, that why genes are on and off, how this cell is different from the other cell. These all things are related to epigenomics, the gene regulation. We have a lot of data of epigenetics inside on the databases. Okay, So you can retrieve it by having knowledge of that data, by having statistical knowledge and computer science knowledge. You can start your career in bioinformatics. Okay. Now we do have some biochemistry students also. So how biochemistry is helping, we can how biochemistry student can integrate into or transition into bioinformatics is basically, we can say, drug design. Okay. For example, if you want to design a drug against a potential biomarker, so yes, biochemistry students can do it. They have knowledge of drugs, okay? And after that, just using computer science and different tools and software, they can design a specific compound that can help to, just if we have a potential biomarker, so we can say that yes, it can affect or it can halt the process or the function of that enzyme or that protein. Okay, so yes, drug design is also very much important part of biochemistry. Okay, you can perform virtual screening, you can design a drug, you can perform a lot of analysis. Like for example, if you have 2-3 thousand compounds and you want to check that either these compounds are harmful or not related to the human body, you can perform a lot of analysis and generate hypotheses. Okay, this is related to biochemistry. The other part is the docking and simulations. If we want to check that, for example, if you have two type of proteins, one is mutated protein and the other is wild type protein, you can check that what is the dynamic behavior, the structure changes inside that protein. You can analyze it. You can predict the structure. You can perform a lot of analysis over it, and you can say that just because of this mutation, this protein is unable to perform its function, and you can prove it using simulations and talking that yes, this protein is good enough or it have lost its activity. So biochemistry students deal with protein. So if they have effective knowledge of protein, the biological part, and if they have knowledge of computer science and statistics, they can start their career in structure modeling, docking and simulations, and they can check that how a protein behaves inside a body when it gets mutated. The other part is that, for example, if we have a drug and we want to check that either this drug can just affect the process, the functioning of an enzyme or protein, we can also perform simulations and docking and prove that yes, this drug is a potential inhibitor of that protein. Okay, so these are small examples of that how you can switch into bioinformatics by not leaving your field. I'm not telling you that you are leaving biochemistry or biotechnology or microbiology. You are in your field, but you are integrating bioinformatics into your research. Okay, so these are the three major fields, and by looking into the participant list, I have seen that there were most of the students were of biotechnology part, microbiology part, and biochemistry part. So this was the reason that we do include these examples. Okay. Now, most important thing is the roadmap. How to start? I have seen that a lot of people vote in that specific part, that how to start with it. So I will explain it in a very simple way. If you want to start your career in bioinformatics, build a strong domain knowledge. And I'm telling you, what was the domain knowledge? Biology, computer science, and statistics. Start acquiring the knowledge of these three domains. Okay. After that, if you have effective knowledge of these domains, so then start learning key tools. Okay. I'm not telling you that start with the advanced part. No, no, no. Start with the key tools like Blast, what is Cluster, Omega, Galaxy, how web-based tools are used to analyze the data. Okay, it's just a one-click analysis. Okay, at least you will be having the information of statistics and biology and computer science, but for example, you are not at the pro level, so you can start with the web-based tools. Okay, the servers that are being used to analyze the data. Once you start analyzing the data and understanding the result, then after that, just start dealing with the databases. Okay, that how the data is present inside these databases, how to retrieve it, and how to start processing it. Okay, so this is the third step. First step is building the domain knowledge. Second step is start dealing with the web-based tools. Okay, third is that start dealing with the real data, that by going through the different databases. The fourth is learn programming. Now this is very much important. Programming, we can say that Python, R, and Bash is the backbone of bioinformatics. Okay, and start learning these skills also, because I can say that if we divide, or we can just say that out of 100% of bioinformatics, 80% of the data is being analyzed using Python, R, and Bash, or we can say Linux. Okay, so you should have knowledge of these programs. Okay, so this is the fourth step. Okay, at least going through these three steps, you might have get interested, and then after that you start with the learning or the learning the major programs. Okay, then after that, if you go through these four steps, we'll start exploring and specialization of what is genomic data, what is transcriptomic data, proteomics, structural bioinformatics, metagenomics, AI. Then we will be start exploring these fields also. Because you know that what is biological data, you know that what is statistics, you know the programming knowledge. So then you can just choose your field. Either you want to go with the genomic part or the proteomic part or the AI part. It's all up to you because you have at this level you have a better understanding. And then importantly, apply and contribute, use biological data, different secondary data and data sets and start analyzing it and start processing it and start building some important hypotheses. Okay. If we, I give you some tips to become a bioinformatics researcher, so choose a field of genomics or drug discovery. Now let me tell you, at this stage, you have effective knowledge of bioinformatics. Now you want to start your research in bioinformatics. First of all, you should have a specific field. You should choose a specific field, either of genomics or drug discovery. It's all up to you because bioinformatics have two major fields. The genomic part, dealing with the nucleotide sequences, and the proteomic part, dealing with the products of genomics, okay, dealing with the product of the genes. So these are the two major fields of bioinformatics. It's all up to you, either you want to the genomic part or the proteomic part. After that, select a disease or select a problem. I would say select a problem. I mentioned select a disease. It was just a little bit specific. Again, to be generalized, select the problem, okay. And in biological part, yes, the problem is the disease in simple sense. After that, read latest review. This is very much important. I'm telling you that you should know that what is being done and what I'm going to do, okay, because research that is being already being published, you can just explore it. Don't, I'm not telling you that leave it, but at least you should do some new research. So for that, read latest review papers and create a mind map and start your analysis. Okay. So these are the very major and generalized first step. If you are a bioinformatician, choose the field, select the problem, read the test review to check that either my problem is valid or not and how much your problem should be valid. But at least reading review papers, you will come to know that what is being done at what and what I can do. Okay. And after that, create a methodology and start your analysis. Okay. So I think so you are now able enough to start your career in bioinformatics. Okay. So I think so this was all from my side. And now we will start with the question answer session. If someone have questions, so you can ask. Okay. You can also ask verbally. And if you cannot ask verbally, so just post your queries in the WhatsApp group and I will answer it. The reason why I make it a little bit brief is just because that I want to answer the queries, okay? Because I know that a lot of queries will be there, okay? So you can proceed. Thank you.
Noted. So thank you so much, Sanadine, for this perfect insight into the world of bioinformatics. And yeah, let's proceed ourselves towards the discussion and question answer session. So we have a few questions in our WhatsApp group. So starting with, okay. So my question is from Sir Nadeem and all the sir, I am a bioinformatics student. I am interested in exploring job and internship opportunities in Islamabad and Rawalpindi after graduation. Could you guide me on where I can apply and what roles are available for fresh graduates in the field of bioinformatics in Rawalpindi and Islamabad?
Okay, so if I would say that you can start exploring the NIH, National Institute of Health. They have specific facility of bioinformatics, okay, and a separate field of bioinformatics. You can just go there and just show your interest. There is also NIGAB, I think so, if I'm not wrong, National Institute of,
Yes, sir.
it's also under the collaboration of NIH, okay. If you want to deal with the plant data, so I think so they are dealing with it. So just go officially, email them, or you can just walk in, share your, post your interest that I want to perform an internship or avail an internship, and you can just you can say start your internship there. And I'm not sure that there might be a lot of different other. Just go to the, you can say, the hospital settings like Shifa International. They, I think so, they do have some we can say internship opportunities because they deal with the genomic data. So you can also go there and just explore it. There might be, but I'm telling you that yes, in Pakistan we do have some. We can say that bioinformatics is evolving in Pakistan. Okay, it's not that much in high demand because we do not generate data. Aga Khan Hospital generates data, Shifa International generates data, okay, biological data, sequencing data, NIH generate data. So now people are moving towards that fields or that the generation of the data. So the more the data is generated, so we can say that we will be in need of bioinformaticians, okay. But still in Pakistan, still we can say that we are far way behind the West, okay. So at least just explore it. You will find an internship, inshallah.
Okay, so moving on. And another question from the same participant as well, that what kind of jobs can we do after graduating in bioinformatics? Job opportunities are limited in Pakistan. Could you tell me which domains in bioinformatics have more job opportunities here and which domain I should choose? I am interested in computer subjects and my goal is to get a job in software houses.
Okay, so I think so. Software houses are more, we can say, biased towards the non-living data. Okay,
Yes,
um,
IT and data
yes,
science. Yeah.
exactly. So software house, I think so that there is a no to a bioinformatician, but still the same answer is that still there is lack of opportunities for the bioinformatician, and the only reason is that hospitals are not generating the data because they are not having the resources. But if you are a bioinformatician and if you want to start your career, start with the freelancing stuff. Okay, if you have knowledge, okay, and if you can analyze the data, so just reveal yourself using different platforms. That is, I can do it. Okay, and there are a lot of students in the we can say in China, in Japan, and different other countries that they want us to analyze their data. Okay, so just make your gig and start your freelancing career. At least you can start by from the freelancing part. And I think so in 2 3 years we'll be able enough to generate a lot of data.
Yes.
Okay.
Okay, moving on. So we have another question is, can we switch to a PhD in bioinformatics after doing MPhil in botany?
Exactly. This webinar was about that how to start your career in bioinformatics. If you have done your botany, masters in botany, you do have a lot of knowledge of biology. Okay. But now you just want to, we can say that start investing your time in computer science and statistics. Okay. At least you have a strong knowledge of biology. It's very good enough. It's good enough. And in PhD, you have 4 to 5 years. So I think so. It's a lot of time to gain information and to gain expertise in these two fields also. And then, unless you are having the potential and the interest, okay. So yes, easily you can transition into bioinformatics.
Okay. And the same kind of question from another participant as well is that can we switch to a PhD in bioinformatics after doing MPhil in medical laboratory sciences? So I guess the answer is the
Exactly.
same.
Yes, because you know about the biology, okay, and medical
Yes.
lab technology. You know that how what type of patient data is being generated. So analysis.
Yes. Okay. So the next question is, what are the resources and databases that we can use to enhance bioinformatics skills?
Okay, there are a lot of data present on NCBR, National Center of Biotechnology and we can say information. If I'm pronouncing it wrong, a lot of databases are present, and huge amount of data is present there. Okay, you can retrieve it and you can start analyzing it. Okay, so and it's freely available. Even we are doing it in our in master's level. I have not generated my primary data. I use secondary data to perform my research. Okay, so a lot of data is being present in database. NCPI, SRA, Array Express, European Nucleotide Archive. So I think so some Japan related database is also there. So we can say terabytes of data is present there. Choose your specific data. First of all, choose your research, then your data, retrieve it, start analyzing it.
Yeah. And we have yet another question, but I think you have already answered it as well. Is that should we have lab results or do we require lab results to do bioinformatics research?
Exactly. Lab results. What are lab results? Lab results are the results of the patients. Okay. Medical. It is a medical record. For example, if you are having CBC, complete blood count, it's a biological data. You can analyze it. You can perform statistics over it. You can train different machine learning models over it. Okay. So yes, it's biological data. So you can also perform analysis over it.
Okay. So the next question is, I am a PharmD student, sixth semester. What bioinformatics has to offer me for my career?
Okay.
Basically,
Okay.
for the
So you're
pharmaceutical student.
exactly. So I think so. Biochemistry research answers this question. You know that
Yes.
what are drugs. You know that what are the structures of drugs. You know the properties of the drugs. You know that why drugs are harmful and why they are helpful and these other stuff. You know, an essential, you have essential by, we can say, pharmaceutical knowledge. Okay. Then if you start analyzing their drugs over the computer screen, you have transitioned into bioinformatics. Simple. How the drugs enters and how the drugs behave inside our body, you can check it virtually. How they interact with the proteins, you can check it virtually by using simulations. So yes, you can easily transition and you can easily start your career in bioinformatics. You will be not leaving your field. You will be in the pharmacy field, but you just have started some advanced research. Okay.
Okay, so the next question is, I'm doing PhD in biochemistry area of specialization is biochemical neuropharmacology. And I'm doing working on addiction, memory, and stress. So how can I use molecular docking? I want to add bioinformatics in my paper thesis work.
Okay. So what was the problem?
So the user is
So,
doing molecular docking on
uh,
the studies of addiction and memory and stress. So they also want to add bioinformatics in their paper thesis work. So how can they integrate
okay,
their
so
work on pharmacology?
if you're okay, okay. So I think it's a brain condition. We can say that.
Yeah.
Just find out that why, what are the causative agent, what are the proteins that are we can say that responsible for this. Okay, what are the pathways, what are the proteins, the pathways, the biological processes that just make the individual to become this such type of or to generate these such type of effects. Okay, after knowing it, the proteins, the biomarkers, you can start analyzing it. You can start just we can say applying different drugs over it. That if you find out that yes, this protein is responsible for this action and you want to just we can say inhibit its function, so yes, docking helps a lot. You can check that what can be the compounds that can halt or we can say inhibit the function of that protein. Okay, simply. So what will happen that docking helps here, simulations help here. Okay, so problem is the same. You're dealing with neuropathic or we can say psychological issues. You find out the biomarker, you apply docking. So I think so this clearly answers the question.
Yes. So a student of microbiology in from Fata University is asking, what do you suggest to me for masters?
Okay, so if you're microbiology students, I would say that start with metagenomics, okay? Because it's we can say that you are analyzing the genomic content of bacteria, so we can say it's completely reflecting microbiology. There are a lot of options also, but metagenomics is better enough. At least you will be in your field. You will be analyzing the genetic content of the microorganisms, okay? And masters there. I myself have done research in metagenomics, being a microbiology student, okay? So at least if in the thesis or in the defense, someone asks you that how you are dealing with microbiology and you have started with bioinformatics, you can easily say that I have deal with the genomic content of microbiota, or we can say microorganism. So start with metagenomics.
Yes. So the next question is, how is artificial intelligence improving bioinformatics research?
It's an important question that how AI is helping us. Help, we can say that uh, at least if you are a new bioinformatician and you want to learn bioinformatics, so AI can pace up your research and your journey. Okay, you can start learning Python, Linux using AI, the very basics of it. Okay, that what is the code, how to process Python, how to use Python. So these are different parts that can be easily understand or easily gained by using AI. Okay, rather than we can say. Going to some physical classes also. AI is helping us to pace up our journey of for being a bioinformatician. Okay, so yes, it's helping out.
Okay, guys, so you can also just ask the questions by turning on your mic and directly ask the Nadeem as well. Okay, so moving on to the next question. From where I get real data for practice because no one is free to share their genomic data.
There is a lot of data present over the databases. The European countries have provided. Okay, so no need of asking someone to give her his or her data. Just go to NCBI. There is a lot of genomic data present there. Go there and download it. Okay, no need to ask someone. And use NCBI, European Nucleotide Archive, SRA. Sequence Read Archive, or you can say Array Expression Atlas. These are the databases. Just go there, retrieve the data, start analysis, perform research.
Yes. Okay. Moving on. So the question is, if a job interviewer asks that why do you want to switch your field, like moving from biotechnology to bioinformatics or any other fields like maybe biomedical sciences or biochemistry, how to answer such questions, like how to make the answer more specific and not a generic one.
Yes, honestly speaking, if uh, we can just say that due to resources, I cannot pursue my research in biotechnology. But I tell you that in Pakistan, just other than some 2 3 universities, you can just perform, we can say, PCR. You cannot perform sequencing. Okay, so just because of the limited resources, you can answer that I have shifted to bioinformatics. I want to analyze the genomic data, but in my research I cannot generate it, but at least I can analyze some other data. Okay, so this was the main reason that yes, I have shifted to bioinformatics to just explore new ends of biology, and bioinformatics was helping me out in this. Biotechnology was not doing it, not helping me out because I cannot generate the data. So limited resources.
Okay, the next question. In disease, for example, cancer, how bioinformatics can be integrated into treatment of cancer?
Sorry, can you repeat it?
So basically, how bioinformatics can be integrated into the treatment of cancer or cancer studies.
Okay, treatment of cancer. If I could say that if you want to find out that why this cancer was caused, what were the culprits behind it, what were the proteins that were making it to cause the cancer. So there are a lot of pipelines to find out, and most importantly, after your research, you can at least generate a diagnostic hypothesis or a therapeutic hypothesis that yes, this was the main culprit. Diagnostic hypothesis, these are the drugs that can be applied to just block this biomarker. This is the therapeutic approach. So yes, treatment of cancer is a major field of bioinformatics to deal with.
Okay, the next question is, which bioinformatic tools are best for detecting antibiotic resistance genes in bacteria and how do they work?
Okay. Recently, our lab developed a tool of ARG Prism. It was used to detect antibiotic resistant genes inside bacteria. Uh, there are a lot of tools. I think so. I've just forgot their names. Just search it out in the Google. At what are the tools? Most of the tools are Linux based, so at least you should know that how to process it using Linux. Okay, because these are advanced tools. We can say that yes, there are tools that are web based tools, can be used using mouse clicks. Okay, um, but sorry for that, I just forgot the names. Okay, but at least ARG Prism, we can say that it is used. Explore it using online resources.
Yes. Okay, so we have Dr. Asmat Malik with us, and he's postdoc in clinical medicine from China. And he's asking, sir, my major is clinical medicine, and I want to integrate bioinformatics into my field. How can I do that? Furthermore, I want to explore bioinformatics. If there are online sessions or hand-on training opportunities, please recommend it.
Okay. Clinical medicine, it's a diverse field. I would like to know that which part of it you want to integrate, you want to use bioinformatics for it. And the other part is that Omics Nexus do provide these such type of trainings. Okay, our organization is the main motto is bioinformatics accessible to everyone. We have free webinar sessions for we can say that for the awareness purposes. And yes, we do launch some trainings, 3 days trainings, 1 week training, 2 weeks trainings related to bioinformatics. And yes, we are releasing some important trainings in the near future, like the bioinformatics summer school. We will be in touch with the students for 2 months, and we will be covering important modules in it. Okay, so you can join the summer school. Recently, after 2 3 days, we'll be announcing single cell RNA-seq data analysis training workshop. It's a 3 days workshop, and single cell is the top notch skill uh, in bioinformatics. Okay, that how to interpret the gene expression data generated from a single cell. So you can join the training. We do provide a lot of training, and we on just visit our website. We do have some trainings. The recordings are there. You can go ahead, explore the syllabus. If you are interested, just mail us. We will provide the trainings, the recordings to you. Okay. So we have a lot of stuff in mind. We already have done it, and we are doing some important stuff in the near future. And just follow our pages. You will be notified, inshallah.
So another question from the fourth semester biotechnology student from US. It's basically one of my juniors. That what basic bioinformatic tools should I learn to be eligible for internships in Lahore?
Okay. By basic tools. So I think so. You have I
Yes.
just mentioned it in the roadmap. Okay, there at
Yes.
least you should know about the web-based tools, the Galaxy, how to process Galaxy, uh, how to perform alignment, multiple sequence alignment, how to design a primer, how to we can say there are a lot of basic stuffs in bioinformatics to start with. Um, some important statistical tests you should know about it, or some web-based tools that can analyze gene expression data, like we can say GO2R analysis. You should know about it, and protein docking. Some
Yes.
it should be web-based, but how to perform docking and um, a lot of other basics tool, but start exploring it. You will find a lot of. But multiple sequence alignment, primer designing, docking and simulations, GO2R analysis, Galaxy, gene expression analysis using Galaxy. These are the basic things, web-based tools basically. And then if you want to
Yes.
transition into the advanced, just go for the internship. Get in touch with us in our webinars and workshops. Then you can just explore the other portion also.
And as far as I have experienced, the things like in the labs, what they're doing projects in Lahore, so they're not like actually working on the big data analysis in which you would have to be the high processing computing or not. You don't require that. So basically, familiarize yourself with the tools like they're using Snapgene, like for the recombinant vector designing and viral primer designings and the Mega 11 tools. So yeah, the basic cluster of tools, and you will be okay with the labs in Lahore. Okay, so moving on. We have master students in molecular biology and integrated bioinformatics. And the question is basically, he's saying the drug discovery approach is in my research. And my question is after studying interaction of our compounds against a specific protein, which is mutated in a certain disease, and after drug likeness and ADMET analysis, what's more to do further? Like it's in silico drug discovery approach, like after this in vivo trials and all come. Basically, the question is not
Okay.
structured at all, but
I understand it. I understand it well. That what he want to ask is that yes, he have just checked the interaction analysis. He want to explore that how his mutated proteins interact with the compounds. Okay, so yes, he have checked that yes, there are certain interactions of the amino acids with the drug compound. Now he also performed ADMET analysis to check that yes, this drug is biologically effective enough or not. We have done it, but most importantly, I would say that go with the simulation part. Check that these interactions are stable enough or they are just, we can say, random interactions. And simulations helps you to just, we can say that, prove those interactions that either they are reliable or they are just random. So what inside the simulations, all the protein, your mutated protein, your compound that is being docked inside the mutated protein, it gets dynamic, okay? Completely dynamic. And after that, using the dynamic rules, if the interaction exists, okay, then you can say that yes, our mutated protein can interact with the compounds. And if it breaks apart, so you can say that yes, our mutated protein cannot interact or cannot withhold the compounds that are docked inside it. Okay, so simulations are the last is the last part. Check the effect of the mutations over the drugs.
so some of our participants are raising hands. So please go on and turn on your mic and go ahead and ask.
Hello, good afternoon. My name is. Can you hear me?
Yes, we can hear
Can
you.
you just
Yeah,
speak a little bit loudly?
my name is Kingsley Ejim from Nigeria. I'm a biochemist, a lecturer, Federal Polytechnic to do that in Niger State. My master's is on cytotoxicity. I'm working on cancer, precisely breast cancer, which I have done my cytotoxicity, I've done my molecular docking, But the last aspect of it is what I'm trying to do, which I find it a bit difficult on a simulation, molecular dynamics simulation. I have done my postdocing using ADMET to detect my drug likeness and the leather LED, the leather dose of my compounds. So, but what I want to ask is, how do I get this software for molecular dynamic molecular simulation so that I will be able to finish part of it? Another question is on the I want to also look at the proteomics and the genomics, the genes in this my breast cancer that I have worked with. I have worked on, sorry, to understand the genes and also to know the nucleotides in these genes and also to know the proteins that the mRNA of these genes has translated. So if I can have the software so that it will help me to move on and on how to operate those softwares. Finally, it's a good journal that I can publish this my work because I'm looking up to see my work in Scopus. So these are the questions that I have. Thank you.
Mm. Okay, so thank you, Ijim, for joining the session. Honestly speaking, I just don't get entire of your queries. It would be better enough to just approach us using our website, but at least most of the things I heard it that you want to perform simulations, and either simulations can help you out in your publication. So yes, I could say that simulations is the last part to just verify that yes, your docking was stable enough and how your compounds were interacting with your protein. So there are a lot of other analysis inside the simulations that how hydrogen bonds are being formed with the protein, how the amino acids are interacting with your compound. So yes, integrating the simulation step can help you in publishing in your research inside the Scopus. I think so. You mentioned Scopus. Okay. The other part of your question was you want to also analyze the genomic content. So I could not hear it very clearly. So I would request you to just go to our website, post all your queries there. I will answer it in an email. And if you want to pursue or you want to have a free consultations, I can just get in touch you with the experts of the field of simulations and the genomic. Okay. So sorry that I couldn't answer all of your queries, but I show you the way that how to just get in touch with us. Thank you.
Yes, thank you very much.
Uh,
I can in the after. Thank you very much, sir, for your explanation. I'm highly grateful. I'm
Okay,
so
thank
much
you.
happy
Yes,
to. So I will
thank you.
speak on a WhatsApp group so that I will get more contact of you for on that simulation and the proteomics and the genomics because these are the three basic things that I want. And where the good journals like Scopus
Okay, sure.
where I want to see my work. Thank you very much, sir.
Exactly, exactly. Good luck for that, and just go to our website and just post your queries there. Okay. Thank you. Yeah, okay, Temur Sikandar, go ahead.
Thank you.
Okay, so any other questions from the audience who have raised their hands? Please
yes.
go ahead.
Hello, Assalamu alaikum, everyone. Assalamu alaikum, Nadeem
Walaikum
sir.
assalam.
This is Rizwanullah, and I'm field scholar of microbiology from University of Sawabi, Khyber Pakhtunkhwa. My question is that once we successfully sequence a mutant gene and determine its protein structure, comparing it to the wild type using bioinformatics tools, what are the most immediate clinical or therapeutic benefits we can expect? Specifically, how does this structural insights directly accelerate target-based drug discovery or remediation strategies? Thank you.
Okay. Thank you very much for getting into this event, Rizwan. Uh, if you have performed the sequence analysis and you have compared your wild type and mutant type, I would just want to ask that have you performed the structure part also? Have you generated the structure of the wild and the mutant part?
Yes, sir. That's my question. That when I generate these two structures, then what could be the
Mhm.
possible therapeutic benefits we can take immediately using bioinformatics tools in solving this problem clinically?
Okay, so okay, the mutations that how my answer is that when you have just generated the structures, so compare that how the mutations have affect the structure of the protein. Okay, one part. The other part is that you should know that what are the important ligands or the we can say the compounds that were used by this protein. Let me explain it in a simple way. You just mentioned bioremediation. Okay, so we can say that there might be some compounds that were effectively processed by the wild type. Okay, so while the protein get mutated, so you can just prove it that because of that mutation, how the compounds are interacting with that protein. Okay, so you can just go with the extended simulation part, that the mutation, how the mutation have affect the protein, and either it is functional or not. Okay, and the other part is the case of bioremediation. If you want to check that how the compounds after mutation are interacting with the protein. Okay, and in comparison, as on a reference, you also check the wild type also. So yes, you can prove that because of this mutation, how the compound interaction was affected, how the structure of protein was affected, and either this mutation have caused to the protein to dysfunction or not. So these are the steps. There might be a lot of more steps, and if you want to get more detailed insights over it, so yes, we have structured bioinformaticians in Omics Nexus. Just go to our website, post your query. We can get in touch, get you touch with structured bioinformaticians. Okay.
Thank you, sir. Thank you, sir. Inshallah.
Okay, so moving on. A question from Asma Rafiq, who is unable to turn on her mic. So she's asking, what are the best public sector institutes you would recommend a biochemistry graduate who's learning Python and Linux to solve biological problems, interested in NGS analysis as well? And is it really worth it to select just one field, as mostly bioinformaticians have all the skills, like gen transcriptomics, in silico drug, pipeline development? And do we really need to learn all before selecting our MPhil research topic? Lastly, is doing MPhil in biochemistry with bioinformatics integration, I mean, acting like a hybrid scientist, is better and practically possible in Pakistan than doing MPhil in bioinformatics alone? So basically, she's asking if the hybrid degree, like integrating bioinformatics into biochemistry, is possible and is worth it in Pakistan than just doing MPhil in bioinformatics.
I'm telling you in the very start and in the event, I was telling you that I'm not telling you that just go and start your degree in bioinformatics and just leave your field. Be in your field and start learning bioinformatics, and using your research interest, integrate bioinformatics into it. Okay, you will be a hybrid scientist because you know that what is biochemistry, and then you have start learning the engineering part. Okay, so yes, being a biochemist, you can be a better bioinformatician. Okay, if you want that. The one thing that if you want to go specifically to the bioinformatics part, I will not just stop you that do not go for it, okay? Because I was also bachelor's in microbiology, and when I start learning it, so I feel that if you I want to get advanced knowledge of bioinformatics and just learn the foundations of it, so I switch my degree to bioinformatics. Okay, two things: if you are in your master's level and you already have started your career in biochemistry, okay, so parallelly start learning the bioinformatics part also. Or if you haven't started your biochemistry, okay, so start your career in bioinformatics, choosing the very specific degree of bioinformatics. Simply, okay. So inside the bioinformatics, you will also learn biology. There are courses that will help you to learn biology, okay. So both the conditions, you can go with both the part. And I have shown you the conditions that either if you have started your career in biochemistry, do not switch it, just start learning parallelly the bioinformatics part. And we are providing the different, Omics Nexus is the platform that provide effective learning opportunities of bioinformatics. Okay, so no worries, you can easily go with each part of it.
Okay, so with the next question, is it a good idea to do cold outreach to researchers as an intermediate bioinformatician with a goal of contributing to real world projects? How open are they to this type of collaborations? And any other suggestions for building a good bioinformatics portfolio would be appreciated.
I won't just repeat it again. I think so. Your voice was not clear enough.
Okay, so sir, the student is basically asking if the cold outreach to the researchers abroad or national is like significant in the research projects or not.
Exactly.
In
You
case
can
of
just
bioinformatics.
exactly. There are a lot of researchers, core bioinformaticians. You can reach out to them. I'm not telling by the just we will mention our Omics Nexus. I have a perfect team of bioinformaticians. You can easily get access to it. Okay, you can easily get in touch with them. I have structured bioinformaticians. I have bioinformaticians that are better enough in NGS data analysis, automation, pipeline, tool development. I have bioinformaticians who know that how to design a project. Just go to our website and book a free consultation, post your query. We will find a better bioinformatician. We'll connect you with them personally and just ask your queries, because all of our bioinformaticians have more than 4+ years of experience in the field, in the real field. Okay, and we have analyzed a lot of data. So no need to worry. We are here for you. Thank you.
Yes. Yes, please.
Hi. We can also consider those who are also here. Please unmute yourself and ask questions here directly. Let's proceed with Saida Fatma Roshan Ali.
Are you here? Hello, sir.
Ji, I can hear you.
Okay, sir. I have one technical questions that which bioinformatics tool are most commonly used in the research laboratories and embedded students?
Okay, Bilal Ahmed, it totally depends upon your research interest. If you are dealing with the protein designing part or the genomic part, so there are different tools that can be used. So you must specify your research area or your research interest. Just share it. We will answer it.
So the question is how to develop a project on thalassemia using bioinformatic tools. Which steps are most important to complete a project?
Okay, thalassemia. I think so. First of all, a lot of work has been done on thalassemia. We do know that what is the causative agent of thalassemia, what type of mutations are there that makes a person to be thalassemic. So I think so. You should start with some literature review and check it out that what portion of the research should be conducted on thalassemia. I have not done a lot of work on thalassemia, so I'm not that confident enough to tell you that which part is should be analyzed. So importantly, just go to search out some literature and check that which part is remaining and how bioinformatics will help me to deal with the thalassemia.
Sure. Okay, so another question is metabolomics the right field to focus, like right after the bachelor's, or should I focus on transcriptomics or genomics rather than metabolomics?
Totally depends upon your research interest. Metabiomics is also very much diverse. Okay, you are dealing with the metabolites. Genomic is also have a lot of potential, and similarly transcriptomics. So it totally depends upon your research interest. Okay, that which part you want to analyze, because these are different omic data: transcriptomic, genomic, metabolomic. So it's all up to you. They all are advanced level of omic data. How much questions do we have?
So we
Timur?
still have a number of questions, like a lot, mainly like 10 to 11, I can say.
Okay, so we'll be answering the questions till we can say 4:55, and then we'll be
Sure,
winding it up. Okay, just
sure, sir.
proceed.
Okay. So kind reminder to everyone that if you're not unable to open your mic, just like write up your question in our WhatsApp group so that we can ask away. Okay. So the next question is, I'm a biotech student. If I want to pursue a master's in bioinformatics, which international universities should I consider that also offer scholarships? And what criteria should I fulfill as a biotech student to get admissions easily?
I would recommend to just explore Google for it, because I'm not in Pakistan. I can tell you Nast University, Qaid-e-Azam, and Comstate University, Islamabad. But I'm not sure about the international universities. Okay, it's better enough to just search it out.
Yes. So I can still answer that because I'm also like researching on the scholarships and all like in these days. So there is a MEX scholarship open these days from Japan, which are offering like programs in bioinformatics, biomedical engineering, and biotechnology as well. And there are also number of scholarships in European countries are open, which are fully funded and like partially funded as well. So go check out the websites like Opportunity Corner or the Scholarship Wizard, and you will get to know more about that. Okay. So the next question is, how can we make a drug in silico to target, for example, an enzyme that have been upregulated during a disease?
Okay, you want to design a drug. So a lot of ways are there. Uh, First of all, why we design a drug This is very much important. That at least, for example, if that enzyme is already having a drug that is being processed, or it is having a ligand that can easily interact with that protein. So using that data, that existing data, you can design your own drug. But at least you should know that why you are designing it, okay? Why you are adding different functional group that can enhance the activity of the existing ligand or existing compound. So designing is easy. You can design a drug, but at least you should know that why you are designing it, okay? And yes, you should follow some a variety of rules to design a drug. Okay, I think so that this theory might not help you out, but it does do have a very span type of answer. Okay, so we would recommend that if you are really interested in this field, you can just meet our structured bioinformaticians in Omics Nexus. So just post your queries on the website.
Okay. So a participant has asked that I'm willing to learn R and Python, but need a teacher as I am biology student and don't know ABC of coding.
Okay, so we do have announced, or we'll be announcing the summer school, and we have a detailed module of the basics of Python, R, and Linux. So just join our school, summer school. It will be started. We'll be starting it in July and August. So you will be with us for 2 months, and yes, we'll be giving you a lot of projects, assignments, and we'll be get in touch with the core bioinformaticians. So yes, we can help you in your journey.
Yes. And yet another question from Riba Iqbal that being a biochemistry student, I am planning to conduct a research project on mitochondrial dysfunction in colorectal cancer. I also aim to study the HMGB1 protein to investigate the molecular damage mechanisms involved in colorectal cancer progression. Along with wet lab work, I want to integrate bioinformatics approaches into the project and ultimately publish a research paper. Could you please guide precisely how I can accomplish it?
Okay, first of all, if you are dealing with the mitochondrial data, so you should have it, okay, related to the colorectal cancer. So the genomic data that is being used by in your research, it should have mitochondrial DNA also, okay. So after getting that data, you can start your analyzing. There are a lot of steps to analyze the mitochondrial data, and yet when you get a lot of findings, so then on the basis of the findings, you can proceed by following your objectives, okay. First of all, what you have to do is select the data that have mitochondrial genetic content inside it. Okay, just go to the protocol, search it out that yes, do they have considered that data or not, and do they have generated it or not, and after that you can start your analysis. Yes, you need a precise answer, and yet this is a very important research question, so you can just reach us out on the website. Okay, why I'm telling the some of the students are asking very much important and very much authentic questions, and I need that they should be answered in a very better way. So the most important thing is that just go to our website and post your queries there. At least
Yes.
you will get the best answer, inshallah.
Okay. So, sir, I guess we have the last question. And if I have missed any, please check out our WhatsApp group and tag those before 4:55 p.m. So the question is, in drug discovery, when we follow YouTube videos, most of them use outdated software and applications. Could you please arrange a workshop? Uh, Let me just, I guess I have lost the question. Yes, so could like you please arrange a workshop where we can learn about the latest tools and applications used for drug discovery, molecular docking, and molecular dynamics simulations?
Yes, though I'm not sure about the YouTube videos, but in our summer school, we do have a module of docking and simulations, and we are planning to use the advanced tools. Okay, so just go reach us out on website, check the syllabus that what we are going to include in it. Okay, and we are planning to include the latest tools that are used for docking and simulation. So hope to see you in the summer school.
So yeah, I guess that's just for the questions. And if you have any other queries, please go ahead and turn on your mic before the time. So go ahead and ask.
I think so. All queries are answered, and just wrap it up. Sikandar.
Sure, sir. Let me just share on my screen. Okay, so at last, what our organization really stands for, let me tell you that if you're really hesitant to step into the field of bioinformatics, that if you have a complex biology fear, or you're afraid of coding and programming, which is the backbone behind bioinformatics, or you have the fear of statistics, which have like a total background of maths and everything, because biology students don't actually like pursue or opt for the math. So in this case, we have actually got your back, because we can support you 24/7. You can like register in our free consultation services as well, and we can guide you through your career path, what you actually require in transitioning or like advancing your career in bioinformatics. We have, like, as I have like discussed earlier in the introduction, we have also conducted webinars and workshops for the beginner level students, and we will be continuing to do so through our trainings, workshop, and crash courses. And we will be helping you with your project developments if you're a researcher in your MPhil and PhD as well. And of course, the best part, our one-to-one mentorship with our experts in bioinformatics. So be confident, because we got your back. Okay, so lastly, before leaving, please follow and check our social media channels, and do follow us, add subscribers on YouTube as well. Do check our WhatsApp channel, Facebook, Instagram, Twitter, and LinkedIn, and do scan the QR for our website and put in your email, and be tuned for our latest information on upcoming webinars. Like Sanadeem has also mentioned, we are starting Bioinformatics Summer School, in which we'll be dealing with all the basics bioinformatics or a biotechnology or MLT or microbiology student would be needing to actually address their needs of transitioning into the bioinformatics or advancing their career. So yeah, Sanadeem, if you have anything to add on, please do. So we can.
No, thank you very much, Emur Sikandar. But I will just mention that Omics Nexus is not just a company; it's a helping hand of the students. They do have a motto, as I have mentioned again and again. We do provide and make bioinformatics accessible to everyone. Yes, we perform free webinars. Okay, we do have a purpose for it, to educate the students. And then after, if you want to learn the advanced level and the complete end-to-end pipelines of bioinformatics, so yes, we also do provide in a very effective price plan, because we know that you all are students, okay, and you have expenses also. So Omics Nexus, what they consider is that we will be providing different trainings programs in a very limited price, okay, in a very effective price plan. So you can just reach us out. The free consultation services, we want to help the students in their research, okay. So 30 minutes of free consultation is also provided. On our website, there are also a lot of recorded sessions of top-notch bioinformatics pipelines. If you want to get access to it, just reach us out on our website. We will provide it to you. Okay. So let's make bioinformatics accessible, and let's make our research more attractive. And I'm telling you that being a bioinformatician, you can easily stand in front of a European student, okay? Because they are also having computer, you are also having computer, and your research can easily be on a level with them, okay? I'm not telling you in the wet lab part. In the wet lab part, yes, we do lag a lot. But in the bioinformatics part, if you are a better bioinformatician, you can stand in front of them. You can do more research, you can generate better hypotheses, and yes, you can explore or deconvolute complex diseases. Okay, so be confident and just think out of the box. Okay, because a lot of opportunities can open when you start with the bioinformatics. Okay, and we are the real faces of it. We can tell you that how we are succeeding in it and how we are processing bioinformatics and managing our lifestyle and expenses. Okay, so just get it in it. Thank you very much for being part of this webinar, and hoping to see you in our other workshops and webinars. And hope at least after attending this webinar, I would say that at least you can just talk about bioinformatics with anyone now, okay? And do mention Omics Nexus and follow us, and so that it motivates us, and we'll be bringing you a lot of different important stuff in the future also. Thank you very much.
Yes, guys. So please stay tuned with us for the future events as well. So I guess we have come to the conclusion of our today's session. So take care of yourselves. Goodbye and Allah Hafiz.